In the reserve's South Meadow is testing the impact of predicted rainfall changes on the grassland ecosystem. Blake Suttle, who began the experiment as a graduate student at UC Berkeley, is now a lecturer at Imperial College in the United Kingdom.
I was reading a lot of forecasts for how climate change was likely to affect different ecosystems in California. There are a number of different approaches that use, predictive modeling to gain some sort of prospective understanding of how climate change will impact this species or that species, or this ecosystem type or that ecosystem type. And there's obviously, that's kind of critical information for us to know.
Suttle decided to replicate the predicted rainfall changes in two of the most respected forecasts. Both called for increased rainfall for the Northern California coast. They differed, however, in the timing.
I took the endpoints of each of those model predictions and basically decided to simulate an intensification of our current winter rainy season or an extension of that rainy season into the spring and summer, and look at how each of those scenarios would affect grassland biodiversity and composition. And so what I've done is buried a network of irrigation pipe out in this grassland system, and all of that irrigation pipe leads to a series of valves at the side of the meadow that connect to a 4,500-liter irrigation tank high on the hillside up behind us. And that tank is gravity-fed by a stream so that it's constantly replenished with water.
Suttle laid out 36 test plots across the meadow and randomly applied 3 rainfall treatments. Ambient rainfall plots where no additional watering was done, more intensive rainfall plots, and extended rainfall plots. Once these treatments were established, he began sampling the plots regularly.
Now here's where we really see the value of a protected research reserve. Each of these is a permanent plot, and these plots have been in place with the irrigation system since the winter of 2000-2001. So we're just completing our 8th year of this experiment in a completely protected setting devoted to ecological research. Now inside these permanent plots, I have a number of different types of samples that I need to take. So I have these 1/4 meter squared permanent subplots, 12 of them in every one of these plots. And several times a year, we'll come into these plots and take a series of measurements. For example, this is a homemade point frame made by a collaborator of mine on this project, Meredith Thompson, who was a fellow graduate student at UC Berkeley. Now we come in and we line up those nails with the top of this point frame, and we come and we drop pins successively, one after the other. And as we do that, we ask what the first piece of plant material, what the first species is that the pin hits on its way down, what it lands on, and what it hits at a point 3 centimeters up the pin. With so much detail into plant responses, we also want to know what happens to the things that depend on the plants, things that eat the plants, and the things that eat the things that eat the plants. I measure ground-dwelling invertebrates in the system, things like spiders, beetles, everything that's crawling around on the ground, using pitfall traps. The other group of invertebrates that we want to collect out here are the foliar and flying invertebrates, so the things that are flying against low to the ground and the things that are living up in the canopy of some of the vegetation. Looks like we've got mostly grasshoppers, katydids, a couple of small wasps, and a crab spider.
Suttle's intensive sampling and long-term focus have produced a series of startling results.
The most striking initial feature of the results that we got out here was the importance of seasonality. Basically, you have a system that gets over 200 centimeters of rain every year. A little bit over 80 inches is our average rainfall. If virtually the entirety of that rainfall falls in November, December, January, February, March, and I come out and dump another 2 centimeters every 3 days for 3 months, that's not a very pronounced shift. There are very few discernible differences between these winter edition plots and the plots that just receive ambient rainfall each year. On the other hand, if it stops raining in the middle of March and I come out here and dump 2 centimeters of rainfall on 12 of these plots every 3 days through April, May, and June when it's otherwise very dry, well, that has really dramatic impacts on the abundance of a lot of different plant and animal species out here. So what we saw early on in the experiment was this explosion of diversity and production, 2 to 3 times greater inside those plots than outside.
In the first year, almost all of the plant species benefited when Suttle extended the rainy season, but that soon changed.
The strongest response happened to be by a little species of legume called Lotus micranthus. Now this is a plant that has sort of the special capability of fixing atmospheric nitrogen and turning it into basically plant fertilizer. So this plant harbors in its roots these bacteria that are able to convert nitrogen gas into a plant available form. So the result was this big increase of this one species, Lotus micranthus. Now the plants that are known to benefit the most from nitrogen fertilization in California grasslands are some of the annual grasses. Now these are plants that came over from the Mediterranean. Most annual grasses in California are exotic species. One of them that's a particular sponge for nitrogen is this grass Bromus diandrus, or ripgut brome. The longer-term response was for some of these winter annual grasses to accumulate so much biomass that they dominated the system, basically smothering out most of the wildflowers that are of greater conservation value. So we're left with a system that's basically a blanket of exotic plants that show very little, if any, response to the water addition itself.
As Sorrel continued the experiment, the extended rainfall plots became more distinct, not just from the other treatments, but from each other as well.
Now you see that this plot stayed really green relative to most of the rest of the grassland, and it's largely dominated by this one species called sheep sorrel. Now this is a spring edition plot, or one of those that's received extended rainfall for the last 8 years, and it's the only one where this plant is dominating. If we look at the closest spring edition plot to this one, what we would find is a cover of mosses and this exotic annual grass, ripgut brome. If we went more over, we would see a field of the native perennial bunchgrass. If we went all the way to the far side of the meadow, what we'd see is a carrot relative mixed in with a lot of geranium, a wildflower. So what we're left with 8 years into this experiment, I've shifted rainfall in a highly simplified way, and we're essentially getting 12 communities moving in 12 directions different from each other. It's emerging such that, one species is coming to dominate in one plot and another species is coming to dominate in another plot. It almost seems like chaotic dynamics where there's this very high sensitivity to initial conditions and maybe to slight differences in things like soil nitrogen and other nutrients. What that tells me is that prediction of climate change impacts a priori is far more complex than we would like it to be. It really is going to depend on any number of factors that can govern the trajectory of a given species or a given community type through time in response to something like a change in rainfall. And it probably becomes more complex still when we're talking about not just rainfall changing, but temperature, carbon dioxide, and this is all happening against a backdrop of enormous human modification of the natural world. We need to not kind of neglect this complexity. We need to embrace it. We need to accept that this is how the natural world operates and use that as our starting point rather than our caveat. And then we can begin to generate predictions that are both realistic and reliable.